Hello, everybody. My name's Emily Pietzu, and I'm here with my co-host and fellow genomics educator, Christina Valianatos. And we welcome you to the Jackson Laboratory Virtual Open House. This is our first live event that we have today. I want to thank Dr. Kat Lutz for being with us. She's here to talk to you as a speaker for the COVID-19 theme. Thanks for being with us, Dr. Lutz. We look forward to hearing from you. How are you doing today?
I'm doing great, thanks. Happy to be here and excited to be working with you today.
Awesome. We're looking forward to hearing about the work that you've been doing on COVID. So before we get started, there's a few items that we want to mention to everybody. We hope that you've gotten a chance to watch the opening ceremonies video, but if you haven't, don't worry about it. It's up on the Canvas site, but you might want to take a look at it because there's some helpful details in there for you to get the most out of all the events this week. And we want to let you know that this event is being recorded. And also review the Zoom etiquette document. It's also on the Canvas site, but it's going to give you some more details about things to do and not to do on the live Zoom events. So just a couple short things and then we'll get started. We do have live closed captioning available for all of the live talks. And you can access that by clicking that CC button that's along the bottom of your screen. And we encourage you to use the question and answer form. If you have any questions during the talk, the moderators will collect those and then we'll be able to ask a few of them to Dr. Lutz at the end. Now, during the talk, we want to let you know that if you do ask a question, you'll be asked for your email and you'll be entered to be eligible to win a prize. So if you do have questions later on, or you think of something else that you want to ask, don't forget to submit those on the online question submission form. So I want to give a little introduction on Dr. Lutz. She is the Senior Director of the Mouse Repository and the Rare and Orphan Disease Center here at JAX. Now, this program includes a growing collection of more than 10,000 unique strains of mice that serve the scientific community to model different kinds of diseases. Dr. Lutz has over 25 years of experience in the field of mouse genetics and working with patient foundations. Today, she's going to speak to us about her more recent work, which is on the COVID-19 mouse model and its application to therapeutic development. So now, without further ado, I'm going to hand it over to Dr. Lutz to give us her talk.
Thanks so much. Thank you so much. Let me go ahead and get this presentation loaded, and we will see if we can't get it to a point where you can start to see my screen. And Emily, if you tell me when you're able to,
we can see it.
Viewing, that would
Looks
be great.
good to me, Kat.
Yeah, so you want to probably just go into full screen mode.
I will definitely do that. Thanks so much.
Looks perfect, Kat.
Awesome. So thanks very much. I'm really happy to be here discussing with you today some of the work we're doing at the Jackson Laboratory on COVID-19 research and mouse models. As Emily just described to you, I am the director of the in vivo pharmacology and drug efficacy testing, but I also run the mouse repository here at JAX. And that means, we're primarily interested in getting mouse models at the Jackson Laboratories that are going to be important to the biomedical community for research and for drug testing and therapy designs. And so, of course, when the COVID-19 pandemic broke, we were very quickly interested in getting these mouse models, identifying these mouse models and figuring out, how we can help in this endeavor. So one of the things I wanted to just, touch on a little bit was to basically talk a little bit about viruses. And viruses are part of our everyday life. They are in many ways have molded and help us evolve as human beings from the time of very early evolution. And some of the viruses that you may be familiar with are things like the rabies virus. Oops, sorry, that advanced. The rabies virus, or maybe even things like measles. Certainly aspects of the flu are also viruses that we're really familiar with. Maybe we've all been vaccinated with mumps and measles vaccines. And also, of course, there's the sexually transmitted diseases of HIV and herpes simplex 2 viruses. So viruses have evolved, and in many ways, they're quite opportunistic when it comes to infection and propagation. And certainly some of the more common viruses we're familiar with, we've been able to mitigate some of the negative effects of these viruses. And I think most recently, some of the work that we've done with antivirals and HIV, have really taken that disease from what is a life-threatening and ending disease to one that is completely manageable at this point. So as you're all familiar with, SARS-CoV-2 hit the United States early in 2020 with an outbreak in China in 2019. This is a cartoon of the virus and how it looks. And many viruses are different in the way they're packaged and in the way they're presented. But certainly if you're familiar with the common media and social media right now, you've probably seen some sort of a version of this cartoon. It's a depiction of the virus itself, and I think one of the things that I wanted to really point out with all the viral components, including the membrane proteins and the envelope proteins, the spike protein here is pretty interesting. It sits on the surface of the virus. And this is the spike protein that attaches itself to the human ACE2 ACE2 receptor that's in the lungs of all of us. And so when viral infection occurs in SARS-CoV-2, it's the spike protein attaching itself to our ACE2 receptor in our cells. This is just a depiction of the virus entering the cell and some of the components that I'll point out that I think that might be rather interesting. So here's the virus in the upper right-hand corner. And can you see my
yes.
Arrow?
Your mouse pointer?
Yeah,
yep.
Great, terrific. So this is a picture of the virus that I just showed you. And as I mentioned, it attaches to the cell by this receptor called ACE2. It also has the ability to be helped in that manner with other genes, such as TMPRSS, which is a serine protease, which allows allows the SARS-CoV-2 to enter the cell as well. Once the cell enters, it kind of goes through a releasing of its viral, of its RNA. This is a RNA-based virus. There's some replication that occurs. More viral proteins get manufactured, they get processed, and they leave the cell. And this is a very basic idea of the infection cycle. So to put it in terms of what happens when an individual gets infected with SARS-CoV-2, this is pretty much, if you can think of a viral exposure, the virus gets into the lungs or to the nasal epithelium of the person. And then this infection cycle starts to progress. So one of the things that we're extremely interested in, as I'm sure all of you are, is how can we really defeat this virus, not just by the way we behave, certainly steps of physical isolation, social distancing, wearing masks, making sure that we have good hand washing and all of the things that we do to protect ourselves. Many researchers around the world are working on various ways to attack this virus, like we have so many times in the past. And there are multiple ways in which you can attack this virus and multiple lines of attack. And many of them I'm sure you've probably heard of already in the media. Some are what we call monoclonal antibodies, or even antibodies that have been recovered from patients. These convalescent antibodies are used at times to treat the infection, or I'm sorry, to prevent the infection and to also treat in such a way that these monoclonal antibodies will prevent the virus from entering the lungs. We also have various protease inhibitors I mentioned, and over here on the right-hand side are some of the different mechanisms in which the therapeutics might act on the virus or the cell itself. And so for monoclonal antibodies, they're really going to, target that spike protein on the virus. Others may make it a little bit more harder and a little more difficult for that virus to enter the cell. In some cases, I think we've all heard about strategies on hydroxychloroquine, which inhibits the TLR pathway. This has shown not to be proven very effective in the treatment of SARS-CoV-2, but it certainly is a strategy, as are some of the other components, like maybe remdesivir is one that you've heard of as well. And this is an antiviral that prevents the replication of the virus itself. And then, of course, there are some things that we know don't really help in the treatment of COVID-2, like bleach and some of the other components that we may have heard of in the social media. Now, I think it's important to think about these in terms of the fact that they're experimental. You may have heard of the monoclonal antibody from Regeneron, and of course remdesivir, and then some of the other steroids are also being used. It's very difficult sometimes to conduct these clinical trials, and I can certainly talk a little bit more about them as the time comes. But what we have here are our lines of attack. So all the different ways that we can prevent this virus from either entering the cell or stop it from replicating are the primary strategies for therapeutic advances. Now, why the mouse, and why mouse models for COVID-19 research? Well, as you can imagine, there is a lot that we don't know about SARS-CoV-2 and a lot that we don't know about COVID-19 and the disease. We still have a lot of information to gather in terms of the associated pathologies. You may have heard not just of the pneumonia and the lung associated pathologies, but there's also kidney. There's also what we call a brain fog, because COVID-19 does infect the brain as well. And there's also, post problems with clotting and various other components. And so we still don't know a lot about the pathologies of this infection, but we're trying to learn. And it's very helpful to have a model organism, if you will, whether it's a mouse, a non-human primate, a monkey, a ferret, a hamster. Maybe you've heard of a number of different animal models that are trying to be used to study SARS-CoV-2. But we're particularly fond of the mouse. And one of the reasons is that mice allow you to really do experiments at high throughput. You can imagine, I think, that non-human primates, and while we have non-human primate centers and people do work mostly with toxicology in non-human primates. Non-human primates are expensive. There's not a lot of them. And so as a resource, they're rather limited. And we need things that are high throughput. We need things that are quick and ways that we can help improve the testing of new therapeutics, vaccines, and antivirals. So ideally, if you had an antiviral or a new vaccine, you would want a model organism that you could test first to make sure, A, that it was rather safe, and B, that it was efficacious. And so if you had a mouse model that was able to be infected with SARS-CoV-2, these are sort of the things that you could do to really advance your understanding of whether these new therapeutics would work. They also give us a lot of information about the viral infection themselves. What are the transmission aspects of this virus? And as I mentioned before, just a lot that we still have to learn about the associated pathologies. Now, when we talk about mouse models for studying COVID-19, I think it's important to recognize that on this receptor, this ACE2 receptor that I spoke to you about, this is the receptor. Sorry, let me just back up because my iPad is being very sensitive to touch. And so that ACE2 receptor is where the virus binds to in the cell. And if you're human, there's an amino acid sequence. It's the KEDMK sequence that you can see here, where I have my cursor over. And that's, really good homology to the spike protein, allowing the ACE2, the virus to enter the cell through the ACE2 receptor. There's variations across different species of that sequence, that binding sequence, if you will. And as you can see with mouse, that conservation of those amino acids is not so great. It is pretty good in non-human primates, for example, in the monkeys and the orangutans. So we know that non-human primates are able to be infected quite readily with COVID-19. But for better or for worse, the mouse sequence is very different here, and it has a protective effect. It's different enough that the spike protein will not bind to the ACE2 receptor, and you sort of get this bouncing off, if you will. It's not a perfect match, and it allows the mouse to be protected against infection. And so here you have two mice talking to each other and saying, hey, isn't this great? We can't be infected with COVID-19. But what we do have are genetic engineering techniques. And these genetic engineering techniques allow us to manipulate the mouse in such a way that we can actually take that human form of ACE2 and express it on the cells, on the cell surface of the mouse lung. And actually, this was done back in 2007 by an individual scientist, Paul McRae and his colleague Stanley Perlman. And this was in response to the SARS, not pandemic, but epidemic that occurred back in early 2003, that some of you are probably way too young to remember. And so, SARS was also a coronavirus, very highly related to what we're working with today with SARS-CoV-2, so it's version 2. And what experimenters did and what scientists did back in 2006 was they took a transgene, and this is the humanized ACE2 gene. And what's driving this is a promoter that allows it to be expressed in the lungs of the epithelial cells. And so now what we have is a mouse model that carries this human ACE2 receptor, allowing it now to be infected with SARS-CoV-2. So this was a mouse model that we acquired at the Jackson Laboratory back in February of the beginning of this year. We did a lot of scaling to get these models to the scientific community. And I'll tell you a little bit about how this mouse behaves in response to SARS infection. So this is the mouse model that was generated. And it will be able to be infected with the virus. And here you can see depicted the virus, and it's an intranasal challenge. And so it's kind of the same way that we all are introduced to the virus. And what you can see here is initial body weight. And these are essentially mice that have different doses of the virus. And so we can titrate the amount of virus that these animals are exposed to. And this purple line here is a very high dose. And these are days post-infection. And so what we see with this particular mouse model is that 5 days after infection, these animals start to become very, very sick. And the percent survival, even 6 to 7 days post-infection, is very low. So almost with 100% mortality rate, these animals will succumb to the consequences of the viral infection. Now you can also dose it down, if you will. So at 10 to the 4th plaque-forming units or 10 to the 3rd plaque-forming units, the mice are still infected with SARS-CoV-2. But like so many people in our population, they're able to recover from that viral challenge. Infection does happen, but it clears and the animals recover. So this is a really interesting model for us because it allows us to model the more severe, most severe forms of the infection. those patients who are hospitalized, develop pneumonia and acute respiratory disease. These are the patients that are on ventilators, and sadly, those patients who often do not survive their infection. And at the same time, we can also mimic infection that's relatively mild as well. So it's a very versatile mouse model. And here are just some of the pathology of the lungs that we see in these animals who have been infected. And so these animals will develop pneumonia, and it's also associated with pulmonary inflammation. And that's going to be really important in the upcoming slide that I tell you about. But just like humans, the result of infection is primarily pulmonary disease. So what about things like cytokine storms? What's actually, preventing these individual people who are infected from recovering? So when a person, or in this case, a mouse, is infected with SARS-CoV-2, there's an immune response that happens, like any time that we get sick or are presented with a virus. And what happens upon infection is there's an immune response, and a variety of dendritic cells, activated T cells, monocytes, macrophage respond to the infection, trying to fight it. The problem with SARS-CoV-2 is that very quickly, and in some patients, this inflammatory response can become greatly imbalanced. And so while the immune system is trying to react to the inflammation, it starts to cycle and it can't control the immune response. And so this is what we call a cytokine storm. And instead of the immune system helping the current situation, the immune system gets hyperactivated and actually starts attacking the body, if you will. And this results in multi-organ failure, hyperinflammation syndrome, and death. And this is something that we see in the animals, the mouse models that we have, where a high dose is presented with these animals. So in addition to all the other forms and pathologies that we see in the lung, we can actually mimic these cytokine storms. And we can test these by looking at markers of inflammation in the blood to see exactly what's happening. And for some of you who are also watching the news and looking at some of the therapeutics that are on the market, steroids such as dexamethasone are used to kind of dampen this inflammatory response. And it's not as straightforward as just giving everybody a steroid who comes down with who comes down with COVID-19, because it's a very tricky balance, if you will, about letting the immune system launch a response without dampening it so that it can fight it. But at the same time, you can't let that immune system get hyperactivated. So other things that we're using this mouse model for right now is to study vaccines and also convalescent plasma. And this was a preprint of a publication that came out from a group in Korea. We've actually shipped these mice to individuals all over the world at this point who are, as you can probably imagine, very aggressively testing various therapeutics using this mouse model. And the idea here is that you take a mouse, you vaccinate it with monoclonal antibodies, or, and in this case, it was a vaccine from a cold adapted virus. And then you challenge it with the virus. And the simple question is, do the mice get sick or don't they? And in this case, the animals who did not receive the vaccine got sick, but those animals who did receive the vaccine were not able to be infected in a way that allowed for progression of disease. And so these are just some of the ways that we can, in a very high throughput manner, test these therapeutics. And you can imagine it could be a vaccine, it could be an antiviral, it could be a monoclonal antibody, it could be any number of ways that we can think of in the strategies that we showed earlier to prevent the disease in these. And so I'll end with some interesting components of genetics, because as we move forward and thinking about who gets sick and who doesn't, and I think this is critical question. We certainly know that there are pre-existing conditions and comorbidities that, including age, that will create a circumstance where people who are older, people who have hypertension, maybe type 1 diabetes, and some other pre-existing conditions are going to be much more susceptible to getting really sick when exposed to this virus. But there are other components that we don't know very much about that are outside of that, and that's in our genetics. And so very recently, there have been some genome-wide association studies that have looked at patients who were more apt to be in critical care, more apt to be on a ventilator. And interestingly, in addition to some areas on human chromosome 3, the blood groups came up as an indicator. So those who are more susceptible or more at risk of getting very sick from COVID-19 have one blood type over another, where O is more protective in that regard. So to think of that, your genes could determine whether coronavirus puts you in the hospital. And so these are some of the things that we're also starting to look at the Jackson Laboratory. And there's a lot of folks doing a lot of research here, not only in trying to make mouse models to study things like vaccines and therapeutics, but also to start trying to determine, who may be more susceptible to getting really sick from this virus. And so our goal, as we say, is to have mouse models as diverse as the patients and as diverse as the viruses. So what I showed you before was the K18 model, which is one model on one genetic background, but that genetic engineering can occur on a number of different genetic backgrounds in such a way that we get a population of animals, just like we have populations of people who get COVID-19. And with that, I will like to acknowledge a lot of my colleagues here at the Jackson Laboratory, as well as collaborators. In addition to Nadia Rosenthal, who's our scientific director, who has really spearheaded a lot of the work here at the Jackson Laboratory. And with that, I will thank you for your time and take any questions.
Awesome. Thank you so much, Kat. That was a great talk. We got a couple questions in from some attendees. Mm-hmm. Maybe we can address a few.
Sure.
We've got one question here asking, can researchers get COVID from the mice?
Well, that's an interesting question. When mice are infected with SARS, with COVID, we're in what we call a biocontainment facility. So yes, it is possible for a mouse to transmit. At the Jackson Laboratory, we do not do infectious disease work. So even though we breed the mice, we never have mice on campus that are infected with SARS-CoV-2. That's a special high barrier containment facility, if you will, where that work happens.
Cool. Great question and very cool answer. Here's another question asking, what are the markers of inflammation that are looked for in the blood?
Oh, there are a number of different categories, IL-6 being one, interferon also being others. So there's a whole chemokine and cytokine panel, if you will, that we can look at in order to see if the immune response is being hyperactivated.
Okay. We've got one question here asking, what inspired you to organize and work on this specific, field, COVID?
Yeah, it was interesting. I had a friend, colleague who works here at JAX in China, and he was in Wuhan at the time and under lockdown. And this was back in January when, things in China really started to escalate in terms of the infection and the government, really taking charge of making sure that they didn't increase the spread. It's not that we didn't know about it here, it's just that it wasn't as much of a reality because we really weren't sure exactly. At the time, it wasn't even considered a pandemic. And we just didn't know exactly about what was going to happen with respect to worldwide spread. So that's how I first got interested in it. He was like I said, a colleague, who said, we've got to, this is coming, it's going to get big, and we got to get our arms around it. So then we started identifying the mouse models from the SARS era that we thought could be useful in studying this particular strain of coronavirus.
Really cool. Well, I think we have time for maybe one more question, if it's okay with you.
I can be here as long as you need me to.
You're the best. This one's wondering, how do you know that the therapeutics that work on mice will work on humans?
Well, you don't. That's a really good question, and that's why we try to. The mice are very similar to humans. First of all, a lot of the biology. We're both mammals, and so there's a lot of conservation of pathways and systems. And so, the more you know about the biology and how you're trying to interfere with it, the more you can be somewhat predictive. But the mouse is a model, and it's a model only. And so that's why when we do have information about, the immune system, the mechanism of action of the drugs, certainly things like vaccines are pretty easy to be looking at the biology. Mice aren't humans, and humans aren't mice. And very clearly our immune systems are different as well. So while we don't know for sure, these are the areas that give us some sort of prediction of efficacy, because when you think about the number of therapeutics that are out there right now, you can't do clinical trials in humans on all of them, right? We don't have enough patients to do that, first of all. And second of all, the number of those therapeutics that won't even make it through safety or preclinical work is pretty great. So this is sort of a screen, if you will, to make sure that we take the drugs that have the highest potential for success in clinical trials in humans.
Very cool. Well, thank you so much for your time today, Kat. This was a really interesting talk, and I know that our attendees enjoyed it.
Yeah, I enjoyed presenting it. Thank you very much.
Awesome. Well, that's all for now, everybody. Don't forget to come back later this afternoon for our first panel conversation with the Women in Science group. They're going to present about life after high school in science. That's all for now.